Indeed, I speak for the Center for Structure and Physics of the Genome that we co-direct with Job Dekker. And I'll tell you about our recent theoretical work from the center. And the work was led by Jeremy Owen, who is also here on the call, and so he will help me answering the questions. And so I just want to introduce Jeremy to this community. And the other author is Dina Osmanovic, who is also on the call. Essentially our motivation here was to understand the role of heterochromatin, and as you've seen, as you heard from the previous talk, heterochromatization changes through development, and so the two primary features of heterochromatin, the way we know it, is that it's spatially segregated from euchromatin, and it's dense. It has a higher spatial density, as known from many measurements, in particular from ChromoEMT from Claude Aucher's lab. And the question is why? So how does this denser regions are being formed, and what's the function of this spatial segregation and the function of high density? Okay, let me switch to this kind of spotlight. Okay, and let me see. Okay, so earlier in collaboration with Job Dekker and Irina Solovey, we looked at the mechanisms that lead to formation of denser heterochromatin in mouse cells. And what we found is that the central for formation of spatial segregation between heterochromatin and euchromatin is actually actual interactions between heterochromatic regions themselves. So what drives the spatial segregation are interactions between B compartments, but by itself, that would position heterochromatin in the center of the nucleus, not at the nuclear periphery, the way we know this in the conventional nuclei. And it's the interactions between the heterochromatin and the lamina that positions heterochromatin at the nuclear periphery. So there is an interesting separation of function between the actual spatial segregation and positioning. So that's as far as the mechanism, but this work assumed that the actual sequence of AB compartments is fixed. However, we know that first of all, compartmentalization changes with development. So on longer time scales, on shorter time scales, generally histone marks that establish the distinction between A and B compartments. So the histone marks also change dynamically through the cell cycle. So the question was, what's going to happen in the system if we were to allow now monomers to change their type from A to B? So that was one of the motivation for this work with Jeremy and Dina, and it's posted on bioRxiv. And what we found, the bottom line of what I'll tell you about today is that it looks like one of at least functional roles of spatial compartmentalization and high density of heterochromatin, these two features, is to maintain the epigenetic memory, maintain patterns of marks through the genome, especially repressive marks. It's unusual because we're basically saying this, you need this 3D folding to maintain some one-dimensional information along the gene. So let me take you through this story. So if you just step back and ask, so generally how patterns of epigenetic marks are being maintained, there are textbook answers to this. So the challenge to having this pattern is replication. So during replication, you necessarily dilute the marks at least twofold. That's under the condition of perfect retention of marked histones. And again, marked histones can also be lost. So in this case, dilution can be more than twofold. Then after replication, this pattern needs to be restored. And there are enzymes that come and recognize the mark on one nucleosome and imposes this mark on the next nucleosome. So that's a conventional reader-writer mechanism of reestablishing the memory after replication. So, but the question is, does it really work, or does it really work? Can it really work this way? And again, important component, is to have this enzyme that you can think about this enzyme as an enzyme that catalyzes reaction. So in our language, we would have A regions and B regions, and essentially what this enzyme does, it catalyzes a reaction when an A monomer is next to a B monomer. This A monomer is converted into another B monomer. So you can think about this as essentially a chemical reaction. And the question is, so can this really maintain patterns of heterochromatic marks, or maintain, in other words, this epigenetic memory? So, what do we mean when we say epigenetic memory? We mean a memory in the following sense: that, so imagine there is some external process that imposes these marks first on the chromatin, and then this mechanism itself that imposes the marks is absent. And we're interested in the maintenance of the memory. It's like memory in a computer. You write something on the hard drive and you want it to stay on the hard drive without you constantly writing it over. And the question is, like, for how long can it stay, and can this pattern actually be preserved just by itself through several rounds of cell division, during which you have this essential dilution of the pattern through replication. I call this kind of epigenetic memory in the sense of John Gurdon, who basically in this seminal paper defines epigenetic memory as the ability of cells to maintain, for example, the gene expression, or in this case, we're talking about patterns of histone marks through cell cycles without conditions that induce this particular pattern. So to test this, how the mechanism, to explore mechanisms that can maintain such memory, we need to kind of have set up a model. So we set up a model that we believe is a rather simple model. So it essentially has a polymer of chromatin confined within some volume. So the chromatin itself has A and B monomers, and then B monomers attract each other. That leads to formation of a denser and spatially segregated heterochromatic compartment. And then on top of that, we also have chemical. So that's something that we and others have done before. And then on top of that, you can add an enzyme that can spread the marks. So they're going to be the spread of the marks. So once an unmarked histone, unmarked nucleosome gets in the proximity of a marked one, the enzyme can then transfer, or not transfer the mark, but establish a mark on the unmarked one. And in parallel to this, there is some loss of marks, and the loss can be replicated, but the loss can also be as a function of time. So we model it largely as just a continuous process of loss, but we also consider the case of primarily replicative loss. So another case, in any case, where you can characterize by some rate of loss, L, and some rate of spreading, S. So important is that there is a spreading process and the loss process, and this is happening on 3D folded state of the polymer. And this is not a new model by itself. It has been considered by the labs of Crabtree, Fabien Ardel, Daniel Jost, Cedric Young, David Maranduzo, Andy Spekovitz, and others. So the question is, like, what can this memory, or what are the conditions under this, what are the conditions under which the system can maintain a stable memory of patterns? And different groups ask somewhat different questions about that. One important note is how the spreading operates. So the spreading can operate in 1D, in which case the mark is just imposed on the direct neighbor along the chain, or the spreading can operate in 3D, in which case the mark is established on a histone that is spatially proximal to the one that's been marked and bound by the enzyme. So we'll consider these two cases. First, the 1D case. This is again a well-known result. We just reconfirmed that in the 1D case, the critical parameter is the ratio of the spreading rate and the loss rate. And unfortunately, this system cannot maintain stability. So if you impose some marks and then follow this system for many cell generations, and I will keep showing this diagram. So one line in this diagram is one cell generation, or one moment in time. And then as time progresses, I'll be showing this the pattern of marks. So here, essentially, what you see on the left is that an originally established pattern vanishes at a particular value of S over L after basically one or two cell divisions. On the right, you see a slightly increased S over L parameter, and you see that in this case, the mark spreads like a forest fire through the whole system. And this is characteristic for the 1D spreading. So in the absence of other one-dimensional kind of marking on the system, for example, in the absence of obstacles of any sort, the system will not be able to maintain the memory by itself. So it either loses the marks or everything gets marked. Now you say, okay, this is interesting, but how about the 3D case? So now when the marks can spread in 3D case, in the 3D. So now you need to do a little bit more to define the system. First, what we're going to do, we're going to have a polymer. We're going to impose the marks. We're going to let it fold, and then go into the interface. In the interface, we're going to let the marks gets lost and spread, but we're going to freeze the polymer in space. We're going to say the polymer dynamics during interface for the second can be ignored, because it's much less than the polymer refolding during mitosis. So in the interface in our model, we have loss and spreading of the marks, but we have no polymer dynamics. Then they're going to be at the stage of mitosis, in which case the marks are going to be frozen and the chromosomes will be refolding. And so then we're going to iterate this over many cell cycles. So that's the setup. And the first thing we ask is, like, what's going to happen during one cell cycle? So we just impose the marks and we see what happens to them in one cell cycle. And the surprising answer is that for one generation, you can achieve a very stable memory. In fact, you let the polymer fold, it will form this dense condensate of heterochromatin, and the marks will be remarkably stable in this dense compartment over a sufficiently broad range of the S over L spreading rate over the loss rate parameter. In fact, so it gives perfect memory, and we want to understand why. Why do you get such a beautifully perfect memory? You can even perturb this memory. You can, for example, let it run. Now this is time within one generation, not over many cell generations. So I let the system run. I impose some original kind of a pattern, and the pattern is largely preserved again. So there's going to be some appearance and disappearance of noise, and then I will do something drastic. So I will erase half of the marks, and I will establish half of the marks at random, and let the system propagate, and it will sufficiently rapidly recover the original pattern of marks. And so it's a very stable memory. Not only it's kind of lasting memory, but it's stable to this randomization events, more drastic than even what you would see in a single round of replication. This is again in one cell cycle. We want to understand why do we get this very stable memory. And a very useful analogy was the analogy to the epidemics. Again, this work, as you can imagine, was done over COVID. So we're all reading and thinking about the spread of epidemics. And in part that what helped us realize that there is such analogy. The analogy is to the SIS model, a susceptible infectious susceptible model, when we can constantly get reinfected, and maybe that's the conditions we seem to be living in right now. In this model, the spreading of the mark now being the disease state, is equivalent to the spreading of the disease, and the loss of the mark is equivalent to the recovery. And if you have a social network of social interactions that is similar to this network of spatial interactions between histones, then there is a critical value of S over L parameter such that there is a critical value of the S over L parameter that is related to the number of social interactions. And if the number of social interactions in the heterochromatin, or say in the densely populated area, is above the S over L, and then the number of neighbors per monomer in a euchromatin, or say, for example, urban environment, is less than S over L, L over S, I apologize, L over S, then at these conditions, you're going to have the spreading of the epidemic within the dense part of the network, and you have practically no infection in the less dense part of the network. So this is very similar to what we see in heterochromatin, when there is a dense compartment of heterochromatin and a loose compartment of euchromatin. And we can maintain the mark if L over S is in a particular range. So what do we learn from this? We learn that actually to maintain the memory, the essential component is not only to have spatial segregation of euchromatin from heterochromatin, but also to have a high density within heterochromatin. So heterochromatin needs to be a few-fold denser than euchromatin. And the other interesting conclusion is that it's going to be no memory of this type in euchromatin because it has low density. with that, we say, okay, that's nice. We have high density of heterochromatin indeed. But what's going to happen if I were to iterate this over many cell cycles? So we get very stable memory for one cell cycle, but what's going to happen over many? And when we start iterating this over many cell cycles, the disappointing finding is that you immediately lose memory. You impose a relatively simple pattern of just one marked region in this, say, chromosomal segment, and this pattern is immediately lost and everything gets marked. Okay, that maybe we didn't find parameters where the system is stable. So we tried, and so everything gets marked. We tried different values of parameters, and the parameters here would be, again, the ratio of the spreading rate to the loss rate, and the strength of self-attraction that forms the heterochromatic compartment. And for every value of parameters, we ran the simulation starting with this very simple pattern of one region being marked. And what we found is that there is no region of stable memory, like none. So either you have no memory at all, so the pattern is no marking at all, or the marking is kind of random, or everything gets marked. There is maybe some interfacial region between this, so you can fine-tune your parameters that would give you memory for a few cell divisions, but it's not going to be stable. If this continues, the memory everything will get marked, or the marks will disappear by themselves. So you can kind of fine tune it like balancing a pencil on its tip, but it's not a stable behavior that we're looking to find in this memory. So that was this moment when we kind of paused this as what's going on. So the system that we and others have been studying really doesn't give us a stable memory. So we really need to find some other component that can provide this stability. And Jeremy indeed found this. And it turned out to be a rather simple idea and very intuitive. And the idea being that marks don't spread by themselves. They require the enzyme that I told you about earlier. And if you think about an enzyme catalyzes this reaction. AB is catalyzed by an enzyme. And if you can write it in a Michaelis-Menten type fashion, there's going to be some intermediate state. And then the enzyme is released back and you get the two marked histones. But this enzyme sets the maximal rate at which this reaction can operate. So if you have a limited amount of the enzyme relative to its substrate, the substrate being histones, then it's not going to. Then we can potentially limit the spreading of the marks. So the simple modification of the model was to introduce the limited enzyme, and it changed everything. Because now, if we try different values of the parameters, under conditions of sufficiently strong self-attraction, we get into the regime where the memory is very stable. So in all of these conditions, of a broad range of S over L parameters, the initial pattern of marks is very, very stable. So that was the major kind of insight, again, entirely due to Jeremy, that helped us achieve this stability. You can impose different patterns of marks. Again, they can be memorized for hundreds of generations. There is an interesting phenomenon of kind of error correction in the system. So some regions can occasionally just spontaneously acquire marks, because in some of the cell cycles, the euchromatic region can be close to heterochromatic and get marked. In a few cell cycles, it will get corrected by itself. That's a very nice kind of error correction mechanism. Again, no memory is perfect. So if you run it for thousands of cell divisions, indeed, you see that there's going to be some loss of memory in the form of losing some of these heterochromatic domains or merge of some of the heterochromatic domains. And in fact, the stable state is one big heterochromatic domain. So this is not a perfect memory, but it's a long-lasting memory. And hundreds of cell divisions is a pretty good time scale of which you want to maintain this. In our bioarchive, we also compare the system with experiments where the group of Anya Groth added labeled histones and monitored dynamics of the recovery of histone patterns, and we see very similar recovery. It requires many cell generations to fully recover some of the lost histones due to replicative loss. The model also makes some interesting predictions. For example, that if you ectopically introduce a labeled region, the longer the region, the larger the region, the longer it will last. This assumption that in principle can be tested experimentally. There were some works along this line, but they impose these marks for relatively short regions to argue that sort of somebody should measure this systematically. Another interesting prediction of the model is that isolated islands are less stable than several heterochromatic islands separated by smaller spaces. And there are also interesting predictions of this model related to the cell-to-cell variation. So in principle, if one were to track a regionally marked region over many individual cells, one would see that on average in a population of cells, you would see kind of a reasonable memory of this region. Nevertheless, in individual cells, these patterns would be vastly different, which argues that when the memory were to deteriorate due to, for example, slowdown of cell divisions or changes in the level of the enzymes, individual cells may start kind of losing their memory of the original state, while on average it may appear that the pattern is preserved. So again, single cell mapping of histone marks can also test these predictions. And just to wrap up, so what's the key idea here? And I think the key idea is that the epigenetic memory is preserved in different forms at different stages of the cell cycle. During mitosis, the marks are preserved, but the structure of the chromosome is vastly different. Then during interphase, the memory of the repressed region is preserved in the form of this dense heterochromatic compartment. That allows marks to be changed during replication and then recovered. So the memory is in the form of marks during mitosis and in the form of the folding of the chromosome during interphase. There are some interesting analogies between this system and associative memory in neural networks, and I'll leave this. I'll skip the slides, and so if you're interested, you can read about this in our bioarchive. To summarize, essentially what we found is that compartmentalization helps maintain patterns of heterochromatic marks. So it's not only that the pattern itself folds into heterochromatin, but this folding helps maintain the sequence. But there are three requirements. The first requirement is that you need 3D spreading. 3D spreading has been shown, but there are still very few examples that were convincingly demonstrated. So it would be great if, as a community, we'll continue examining mechanism of spatial spreading of the marks. It requires a denser B compartment. This is something that I think there is a consensus about. So we still don't know what mechanism leads to this densification. And it also relies on a limited enzyme. So that the level of the enzymes that spread the marks should be under strict control. And with this, so we can start thinking about chromosome as a memory machine that helps preserve the one-dimensional sequence of marks or episequence. And I think it's an interesting, for me, it was a surprising finding that 3D folding helps maintain the 1D sequence. And with this, my acknowledgements to Jeremy and Dino, who did all the heavy lifting, to the funding by 4DN and NSF. And again, you can read about this work on bioarchive. And thank you.